Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 152nd seminar, and I call it a University of Minnesota Day. Happy Lunar New Year. I'm very excited to embark again on my 4 weeks of international trips to Spain, Portugal, and New Zealand to give invited talks, attend a conference, and meet people for discussion on global collaboration. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Vincent Noireaux. He is a professor at the University of Minnesota. I have known him for more than a decade through his lab alum, Dr. Xuejun Zhu, who is also Chris Boyd lab alum, although Dr. Sin and I did not overlap. Vincent is a pioneer who studied and promoted cell-free engineering with some colleagues, and I believe many of you are using his cell-free experiment protocols and method. He is also an active leader of the Build a Cell community with Kate Adamala and others. Vincent, thank you so much for your contribution to the scientific community and education, and the virtual podium is all yours now. And thank you again.
Seok, thank you so much for this introduction. Hello, everybody, wherever you are. So I'm going to give a short 10-minute talks about cell-free gene expression, as you see in the title, that I'm going to also call cell-free transcription translation, or TXTL. That's it. So you see a few examples here on the left. I will get back to that in a few minutes. I want to highlight the work done by all of those postdocs and graduate students, and here the sponsors for this work. So let me have this first very general slide about what is cell-free gene expression, or TXTL. You see on top here the workflow. Typically, we work in tubes or well plates, and when you use a TXTL system, you prepare your DNA, you have a TXTL mix, the one you prepare in your lab or the one that you can buy as a kit. And then you mix both to execute your DNA, which can be one gene, multiple genes, and you will see large genomes. And then you incubate for a few hours. It depends on the system that you use. You can incubate a few hours up to 20 hours, and that you get your product or your application. And here you see the 3 categories of applications, what I call prototyping. You can very quickly characterize, get the performance of gene parts, of small gene circuits, of enzymatic pathways. I'm going to show you just after the example of CRISPR, and you can also do unnatural chemistries. The second category is what I call biomanufacturing. You want to produce something, typically a peptide or a proteins. You can also produce membrane proteins. And you will see just after, again, you can produce all phages or part of phages. And then finally, the third category of applications are synthetic cells and interfacing cell-free gene expression with materials. So you can develop diagnostics, microchips, and a bunch of different type of synthetic cells. And that is a snapshot of what the community, cell-free gene expression community, does in just one slide. So my lab developed a cell-free gene expression system that is now sold by this company, Arbor Biosciences. It's called myTXTL, and you can use linear or circular DNA. You can use E. coli promoters or T7 promoters. It is based on a lysate that we prepare from E. coli, and the lysate provides the transcription and translation component, and all of this is very stable at -84 years. You have the substrate, you have an ATP regeneration system, the 20 amino acids, ion and cofactors. It is a cytoplasm that we reconstitute, in fact. Without genome, without DNA, the only DNA is the one that you are going to put in your reaction. And then you have many ways to measure your product. You can use reporter genes. for translation, as you can see here, but also for transcription with fluorescent RNA aptamers. You can do SDS-PEG, you can do mass spec, and many other techniques. So you see on the left what you do. Literally, you take your DNA, you put that in your mix, and get your product. So I'll show you just an example here. We express the T7 RNA polymerase to express GFP, and you see on those curves, the y-axis is GFP in micromolar, and the time is the horizontal axis. The negative control, the blank, is at the bottom in red. And you see the accumulation, the cell-free synthesis of GFP over time. And here it lasts about 10 to 12 hours. And I hold here a tube with GFP at the bottom that has been cell-free synthesized after a few hours. So I'm going to take a metric, which is a size of DNA that you can process into a cell-free gene expression system. You see the log base 10 of the base pair here of the DNA program. Let's start with 10 to 1,000. You can very quickly characterize gene parts. You can express reporters. You can express enzymes, membrane proteins. I show you that example. It's a collaboration with Harris Wang at Columbia University. We prepared 10 different cell-free gene expression systems. E. coli is on top. And we measure the performance, the strengths of hundreds of promoters in parallel. Each vertical line, very thin, is one promoter, and you can scale and measure very quickly the strengths of each promoter. We use for that next generation sequencing. So we couple cell-free gene expression to next generation sequencing. Let's go up a little bit. You can express pathways, gene circuits like CRISPR, do controllers and oscillators. We were the first with SynBYSS to show that you can prototype very quickly CRISPR technologies, not only guide RNAs, but CRISPR enzyme, anti-CRISPRs, and many other things. That's it. And then finally, you can literally express small genomes, and one particular case are phages. We demonstrated that you can entirely synthesize phages in a test tube without a living cell. And the largest that we expressed and synthesized so far is a T4 phage, 169 kilobase pair, 289 genes. Here I'm just showing you the phage T7. You see the particle of the phage T7, and many of the small bullets here are actually the ribosomes. That's it. So let me continue with those phages we are focusing right now. You literally do that like using a PCR product or a plasmid. You take your phage genome, you mix that with your TXTR reaction. Typically, we work in tubes and well plates. You express, and at the end, you have your phages. We demonstrated now. It's just a few that we have done. We have more than a dozen of phages synthesized. You see MS2, 5X, T7, and T4. And the fact is that you can synthesize phages that are not specific to E. coli. We demonstrated recently that you can entirely synthesize phages from other bacteria. It does not always work, but some of them work, some they don't. And most recently, less than a year ago, we came up with this work which is called phages, for phage engineering by in vitro gene expression and selection. We showed that you can entirely reassemble phage genomes very quickly, insert new genes, do some deletion, do some mutation, and then very quickly cell-free express and select. So the workflow is there. You amplify your genome. You have now DNA polymerase that are very fast, and they can amplify very large DNAs. And then you assemble your DNA about 15 minutes. It's a new technique to very quickly assemble DNA, especially large pieces. And then you cell-free express in a few hours, and in a few hours you select, and eventually you can do some next generation sequencing to check the genome of your phage and if your deletion, addition, or mutations are there. Another field of application, especially for my lab, is our synthetic cells. There are three types of synthetic cells: protocells, which is more about the origin of life; minimal cell, which is more about using the real biomolecules to build a cell from the molecular component; and artificial cells, where you are going to have non-natural but artificial components in it. So we just published recently this paper. What you see here are not liposomes. You see droplets without physical membrane. It's a liquid phase separation. We can express GFP, localized in droplets without membrane. And here you see we express the cytoskeleton protein MREB, and it goes at the interface like in E. coli. The minimal cell, you encapsulate your reaction in liposomes. Now, what you see here, we have a physical membrane made of phospholipids, and you see on the left here the expression of GFP, and here the expression of MREB that I showed just before for the protocell. But now MREB is able to deform the spherical liposomes into rod-like shape that looks like E. coli. And then here you have a small movie of the oscillation of the mean system, and the mean system oscillates in E. coli to center the division mechanism. And here it is. No, oh, give me a sec. Here we go. That's work done with Roy Barziv at the Weizmann Institute. It's a chip, and we are able to, in small chamber, to attach physically the DNA on the chip, and we run gene circuits. I'm going to show you an example here of a circuit that makes waves. So let me go there. Here we go. So you see here, each chamber expresses a circuit, a similar circuit. It cascades onto the neighboring chambers, cells, and we are able to create some waves, and we're trying to figure out how to use this for some application. Here we go, and I'm going to close with that. I show you just a few examples of cell-free gene expression. I started with just a snapshot of what the community does. A lot of applications, many new applications are coming up every year, and I showed you a few examples of what my lab has done with many collaborators using this mighty XTL technology, and I name all of those collaborators here that I took some example in the presentation, and that's it.
Thank you so much. It's amazing. I mean, now I see, we have the much more, ability than, 10 years ago. At the time, we only imagine, but you make that imagination a reality. So thank you so much for this wonderful work. And I want to see more in the future. Thank you.
Thank you, Taiseo. Thank you, everybody.
Yep. All right. Now, the main speaker of today with a longer introduction. Dr. Xuejun Zhu is an assistant professor in the Department of Chemical Engineering at Texas A&M University. She earned her bachelor degree from Nanjing University in 2012 and her PhD in chemical engineering from the University of California, Berkeley in 2017 under the mentorship of Dr. Wenjun Zhang, who was a keynote speaker of our SynBYSS conference in Hawaii last year. Following her doctoral studies, she completed a 2-year postdoc at MIT with Dr. Jim Collins, who, of course, a synthetic biology father, before joining the faculty at Texas A&M University. Her research focuses on enzyme discovery, characterization, and engineering. I believe the combining two experience as a postdoc and also graduate student. Her work also involved the design and assembly of recombinant microorganisms to produce small molecules. She has been recognized with several prestigious awards, including the NIGMS Maximizing Investigators Research Award for Early Stage Investigator in 2022. In short word, I mean, this is a MIRA, I believe, one of the most competitive award at NIH. The R.T. McFerrin Department of Chemical Engineering Outstanding Junior Faculty Award in 2022, the Engineering Genesis Award from Texas A&M Engineering Experiment Station in 2023, and the Dean of Engineering Excellence Award from TEES in 24. Susan, thank you so much for your time today, and please take it away, and it's all yours now. Thank you.
Yeah, I was muted. So are you able to see my screen right now?
Yes.
Okay, cool. So thank you for the invitation and giving me the opportunity to share what I've been doing. And thank you for that very long introduction for me. So yeah, today I'm going to talk about what my lab is currently doing at Texas A&M. Basically, we are trying to engineer microbes for a healthier and renewable future. So as we know, there are two major global challenges. One is related to the health. The other one is related to the environment. And for the health side, for many of the drugs that we are using right now, it's becoming ineffective because of many organisms have grown resistant to them already. And for the environmental side, it's been expected that in 30 years, we are going to have about 10 billion of people on the planet, which means we need to produce 50% more of the food and energy we are using right now in order to feed and support ourselves in the future. And it also means that we will have more pollutions and waste during the food and energy production, which may decrease our quality of living. So our lab is particularly interested in harnessing the power of biology, especially the power of the microbes, to address these two challenges. So briefly, my lab has two directions. One is to discover new biology and chemistry. The other one is to use those newly generated knowledge to engineer microbes to tackle those challenges. So I'll first talk about the discovery side, then I will talk about the engineering side in today's talk. So to discover the new biology and chemistry, we are focusing on three different sources. One is from the gut anaerobes, the other one is from conjugated type 4 secretion system, the last is from environmental bacteria. And today I'm just gonna focus on the gut anaerobes and the conjugated T4SS. So we all know that gut microbiota is a hidden organ in our human body, and it's composed of trillions of microbes, and they could be bacteria, virus, or non-pathogenic fungi. And more importantly, all of these microbiota, in total, they encode 3 million of the genes, which is about 150 times more than the total amount of genes encoded by the human genome. With this being said, it is not a surprise that gut microbiota could perform very diverse functions. For example, they could help with the digestion. They could also help with the metabolism and facilitate the development of the immune systems and the neurological systems of our human body. And more recently, people also found these gut microbiota also plays roles in the individual variations of the drug response, meaning when a physician prescribes a medicine to the patients, sometimes the medicines are very effective and it can treat the disease very well. But sometimes the medicine might not be effective or even cause very serious side effects. And of course, there's multiple reasons contributing to these inter-individual variations of drug response. But people recently found actually gut microbiota plays some roles. Here are just some examples. For example, when we take some antibiotics like penicillin, sometimes it's not gonna be very effective because of the presence of enzyme beta-lactamase that's encoded in the gut microbiota genome. And also, when we have some irinotecan anti-cancer drugs, it may cause very serious side effects because of the presence of some bacteria in the gut. And levodopa, that's another medicine for treating Parkinson's disease. It sometimes is not very effective because of the presence of a bacterial tyrosine decarboxylase that's also encoded by the gut bacteria. And more recently, Goodman Group from the Yale University, they found that about 2/3 of the FDA-approved medicines that's being assayed in their study could be metabolized by at least one bacteria strain in the gut, which means the metabolic capacity of the gut bacteria is huge. Therefore, we think understanding the metabolic capacity of the gut bacteria is very important on the health side. And in addition, it could also help us to achieve several goals, like it could help us to develop some personalized nutrition or precision medicines, or even help with the drug development. For example, if we know which genes are important for a certain drug modification, and in the future, maybe the physician could just look at analyze the fecal samples of the patients, and then perform some transcriptome analysis or proteomics analysis and see if that certain gene or proteins or enzymes are present in that patient gut. If yes, then maybe the doctor could prescribe the medicine plus some inhibitor or the medicine plus some cocktails to improve the efficacy of that certain drug and also increase the treatment success rate. All right. But to realize these goals, we need to answer a few questions. For example, first, which specific species are responsible for the compound modification? And second, which chemicals could be modified by bacteria strain? And third, which enzymes are important for such chemical conversion? Since, as I mentioned, there are trillions of microbial cells, and there's about thousands of FDA-approved small molecules on the market. So it would be a lot of combinations for us to test if we want to test experimentally in order to answer these questions. So we think to quickly answer these three questions, it's better to have a high throughput prediction methods. But unfortunately, many methods we are using right now are not very high throughput. For example, many gut bacteria, those are oxygen sensitive, meaning they can only grow in anaerobic conditions. And to do that, we have to culture those bacteria in anaerobic chambers, which is a glove box free of oxygen. But because of the limited space in these anaerobic glove box, it's kind of challenging to perform experimentally in high throughput manner. In addition, if we also want to analyze the metabolic profiles or the metabolites produced by those anaerobes. To do this, we need to have some dedicated analytical instruments such as HPLC, LCMS, or GCMS, which are expensive and usually it's difficult to have access to those instruments. And third, many of the gut microbiota organisms are non-model organisms, meaning the genetic tools to modify those organisms for gene discovery or enzyme discovery is not available yet. So it's kind of hard to deal with these organisms. But very luckily, machine learning recently has emerged to be a very powerful tool, especially in the field of biology, because it could help us to do some prediction and analysis in a very high throughput manner and could analyze large and complex data sets. It could also help us to make predictions automatically, even though we do not have any prior knowledge. And here's a lot of successful examples. Here, I'm just highlighting one. My postdoc advisor, Jim Collins' lab, they have used deep learning approach to discover some antibiotics that's approved to be very effective towards many notorious bacteria pathogens. So motivated by all of these successful examples, our lab was thinking like maybe we could use machine learning to help us to mine the metabolic capacity of some gut anaerobes. So to prove this concept, we selected a specific gut bacteria strain called Clostridium sporogens, which is very representative species in the gut anaerobe. So briefly, we trained molecule X models, that's a machine learning model, with the training data we obtained from the Goodman group and used the models to predict the stability of 280 small molecules. And the top few molecules that's being predicted the most unstable was subjected to in vivo experiments. And later we figured out the enzymes responsible for such chemical modifications. And this project was led by my grad student, Xuejun Zhu, in my lab. All right. And so for the machine learning part, we were doing this by collaborating with Dr. Ji and Dr. Xu from the computer science and engineering department in our university. So after the prediction, we found there are 7 molecules that's being predicted the most unstable, and they all have very diverse, very different chemical structures. They also have very different pharmaceutical applications. For example, everolimus, that's anti-cancer drug being used right now, and the nitisinol, it's a tyrosine metabolism inhibitor, etc. So then we moved on to perform the experiments to validate if those medicines are indeed unstable or could be metabolized by the Clostridium sporogenes. So to do this, we incubate the drug individually with the bacteria of interest. And also in parallel, we have negative controls, which one is the TSB blank medium control incubated with the drug. And the next one is acidified medium incubated with the drug. The reason why we include this acidified medium is because we found Clostridium sporogenes is able to acidify the medium after overnight culture. All right, and then after overnight, the samples were then subjected to the LC-MS to quantify the amount of the parent drugs after the incubation. All right, so from our results, we found indeed a few molecules such as everolimus and a few other medicines could be indeed degraded or modified by the Clostridium species as shown in the third bar over here. And for the esomeprazole and esomeprazole, both of these molecules could also be degraded by the Clostridium sporogenes culture, but the levels of those compounds are also degraded in the acidified medium control, which is consistent with prior literature. For the nystatin, which is an antifungal medicine, we don't see big difference between the negative controls and our experimental group. And then we moved on to figure out what are the metabolites of those parent drugs. So as an example, we focused on determining the metabolites of everolimus by the Clostridium sporogenes. So based on our LC-MS analysis and MS-MS analysis, we figured that the metabolites of the everolimus actually is an alcohol form of the everolimus, in which the ketone group originally over here is converted to the alcohol group after the incubation with the bacteria strain. And then we moved on to figure out what are the enzymes responsible for such ketone reduction reactions. So what we did is we performed bioinformatics analysis to identify some enzyme candidates. And then we purified the recombinant proteins from the E. coli and then incubated the enzymes with the medicines in vitro through biochemical assays. And we found there's one enzyme called EVOB3. It is able to convert this ketone to the alcohol group. And by the way, this EVOB3 is purified as a yellow protein shown over here. That's because it's bound to the cofactor FAD. And then, so here is the ketone reaction that can be performed by the EVOB3. So on the other hand, ketone reduction is also a very important reaction in the pharmaceutical industry. Many of the medicines, they have alcohol group over there. And usually those alcohol group is through the reduction of the ketone group. And for now, many the methods we are using to do this ketone reduction in the industry is through organic synthesis. But as we know, for the organic synthesis, we usually have to use some toxic reagents, and also reaction conditions are harsh. With this being said, it's kind of not very sustainable. So as biochemists, we are thinking maybe we could use enzymatic tools to make these ketone reactions much more eco-friendly. So what we think is maybe we can have a collection of enzymatic tools. They could have a few advantages, like what I said. They could be eco-friendly. The reaction conditions usually is very mild. You can just perform those reactions in the water phase. And in addition, those enzymes could be very efficient and very specific towards the substrates. All right. But in order to have a collection of enzymatic tools, we would want to have a collection of natural enzymes to start with. Then we can engineer those substrate specificities through either directed evolution or rational engineering. But to increase our successful rate of the engineering would also prefer, at least for me, I prefer to do rational engineering. But for rational engineering, we need to understand the mechanisms, how those natural enzymes have certain substrate specificities. All right, so this is an overall picture for this project. So then we looked through the gut anaerobes, and then we identified a few enzymes that could perform the ketone reactions. And those enzymes could be from E. coli, like a KDUD, FABG, those are from E. coli, or some enzymes from gut bacteria, such as Daria, Plautia, Ruminococcus, or Clostridium. And all of these hydrogenase has activities towards ketones, but towards different kinds of ketones. So we tested many different chemicals shown over here, including nabimetone, hydrocortisone, raspberry ketone, zingerone, doxorubicin, etc. And we found indeed these enzymes could convert those ketones to the alcohol form, but the specificity or the activity towards those ketones are kind of different. All right, so this project is overall led by the grad student Liang Yu in my group, who just graduated a few years ago. And then all of these homologs are very similar to each other. The identity to each other is about 30 to 50% identity, but they have different substrate specificity. So we then moved on to figure out why those homologous enzymes are kind of different towards the substrate selection. So we, by collaborating with Dr. Jayaraman, Mittal, and Mohanty from the chemical engineering department in our university, we eventually figured out a few single residue mutations on those enzymes would be very efficient to alter the substrate specificities. For example, if we change the asparagine of the KDUD to a leucine, we could improve its activity towards nabimetone a lot. Whereas if we mutate the isoleucine to Q of this KDUD, we could decrease its activity towards hydrocortisone a lot. And we also found the blue regions highlighted over here is very important for the recognition and conversion of doxorubicin to an alcohol form. All right, so this is our story about how we use gut anaerobes for as a new source of new chemistry. So next, I'm going to talk about we use conjugated T4SS as another source for a new biology. So as we know, antibiotic resistance in gram-negative bacteria is a very serious global challenge. It can cause diarrhea, pneumonia, and meningitis. It's been causing 2.8 million infections and more than 35,000 deaths every year. So there's a lot of infections caused by these gram-negative bacteria. One of the reasons why those gram-negatives are kind of difficult to be treated is because on the cells of those bacteria, there are two membrane systems. One is the inner membrane, the other one is outer membrane. This outer membrane could act as a drug permeability barrier to prevent the small molecule from penetrating into the cells to reach their protein targets. So to address this challenge, our lab has come up with a way, like we propose, like maybe we install a needle on the surface of a good bacteria and use it as an antimicrobial agent. Then incubate this antimicrobial agent with a target bacteria. You could view it as a bad bacteria that we want to eliminate. So once we incubate them together for a while, we hypothesize that the target bacteria could be eliminated or lysed. And the pillars we use to install on this antimicrobial agent is coming from conjugated type 4 secretion system. And this project is led by the student Lois in my group. So in case you are wondering what is a conjugative type 4 secretion system. So this type 4 secretion system is encoded by, is assembled by 13 different genes. And here are the list of the genes that's important for the biosynthesis of this type 4 secretion system. And secretion system, on the structure wise, it is a protein-based nanomachine that's anchored on the membrane of the bacteria cells. All right. So next, we cloned all of those 13 different genes onto a cloning vector and come up with a synthetic plasmid. Then we introduced this synthetic plasmid into an E. coli and use that as an antimicrobial agent. And then we incubate that with different target bacteria. We call them as recipients, and including E. coli, a pathogenic E. coli, Enterobacter cloacia, Klebsiella pneumonia, and also Pseudomonas putida or Pseudomonas aeruginosa. From our study, we found indeed the antimicrobial agent could have some killing effect. For example, this E. coli pathogenic bacteria could be eliminated by about 100 to 1,000-fold through our study. Next, we wondered what are the killing mechanisms. So we demonstrated, for now, lethality is dependent on the contact, but it is independent of the cargos. So briefly, to demonstrate this, we performed a very elegant experiment. So we have an agar plate shown over here, and on top of the agar plate, we spotted a donor E. coli layer, which you can view it as an antimicrobial agent. And for this donor E. coli, it expresses GFP plus or minus the type 4 secretion system. And on top of this donor E. coli layer, we spotted another layer, recipient E. coli layer. And for this recipient E. coli, it expresses D. tomato. You can view it, the recipient E. coli, as the target bacteria that we want to eliminate. And on the note, the donor E. coli and the recipient E. coli, they both have the same genomic background, except the difference I shown over here. So from our experiments, we found the recipients that's in contact with the donors are eliminated. Shown over here, you see it's devoid of the red fluorescence. So from the results, we conclude the lethality is dependent on the contact. In addition, we also conclude from these results, the lethality is independent of cargos, because if there is a T4SS transferable molecule essential for the killing effect, we would expect that the donor should already contain some genes to protect itself from being killed in the first place. Since both donor and the recipient E. coli have same genetic background, except the T4SS carried, the recipient is expected to also have the immunity. And therefore, in that case, it should not be killed by the donor. But from our results, the recipient is still killed by the donor, meaning there's no T4SS transferable molecules essential for the killing effect. All right, so now I'm gonna switch the gear to talk about our efforts on the engineering side. So our lab is particularly interested in engineering the biosynthesis of benzoxazoles for some pharmaceutical applications. So benzoxazoles, it is a functionality, features a benzene ring fused with an oxazole ring. And this functionality has been found in many different pharmaceuticals, which have applications for anti-inflammatory, muscle relaxant, or for treating some heart disease. And this benzoxazoles has also been found in many natural products, secondary metabolites of the SynBYSS. And these natural benzoxazoles have applications in, has bioactivities as antimicrobial, anti-cancer, or anti-leishmania activities. Despite their antimicrobial, anti-cancer, or anti-leishmania activities, their applications for pharmaceuticals has not been widely explored. One of the reasons is the lack of a general and sustainable rapid synthesis platform to then generate a big library of benzoxazoles for drug screening effort. So to address this challenge, what we did is we engineered an E. coli strain by introducing the essential biosynthetic genes into those E. coli, and then we used precursor-directed biosynthesis by feeding in the substrates into the E. coli culture. And afterward, after 2 days of incubation, we found this strategy is able to result in many different benzoxazoles, such as the anti-leishmania compounds or anti-cancer compounds or antimicrobial compounds. And we incorporated different functional group into these into this position over here. And eventually we are able to generate at least 18 different analogs just by these E. coli cultures within 2 days. All right. Okay, so now I have introduced to you a kind of full picture of what my lab is doing. One is on the discovery side. We would like to discover new biology and chemistry. The other one is on the engineering side. And the last but not least, and this is the most important slides in my presentation, I would like to thank all the members in my group, the grad students, the postdoc, and very smart undergrads as well, especially Huanrong Ouyang and Lois and Liang Yu, who have contributed significantly to all of the projects I've mentioned today. I would also like to thank the collaborators. They have given us a lot of help and input when we were performing these projects. And also, I would like to thank all the funding sources that support my research program, and also any questions from the audience. All right, thank you very much.
Fantastic talk. This is a wonderful.
Thank
We learn a lot from this presentation. I wanted to ask if Vincent's still around.
Yes, I am. 10 minutes. Yeah.
Sure. Do you have any comment or question?
I'm good.
Okay, so let me start with the one just technical question. This is about the first part of your talk. This is more interesting to me. This is fascinating. The question I have is now, we could use the machine learning or AI to sort of predict whatever complex behavior in the gut. And then you identify ketone reduction enzyme. And one question I have is, so after this, compound, ketone compound become alcohol, compound, is it still active in terms of the original drugs, efficacy, or this is less active or even toxic to human?
That's a very fantastic question. Many people asked me this question before. Yeah, so at least I have one example here. So there's a drug called tacrolimus. That is a medicine that's to prevent the organ rejection after kidney event. And the people found if they reduce the ketone group to the alcohol group, that metabolites is not effective. So that's one example. But here I showed many other examples, right? But there's not that many in vivo studies right now to see if they are going to be becoming ineffective. But there is one study we found.
I see. So the one suggestion I have to you is you could contact those pharmaceutical company who originally developed these drugs and then do whatever collaboration study, because they will give a lot of money to you, I guess. The thing is, I mean, now I start to understand more, or people understand more things, why some medicine working for this person, but not working for the other one. Why this much, drug kind of having side effect, including myself. I have a huge side effect with some random drug I got, many years But some people is okay with those kind of thing. We may just blame microorganism in your gut, because I mean people has different microorganism, and then those microorganisms do something completely different thing. That cannot be just medicine for treating something. But some people getting that drug that become the toxic chemical or even not effective. So that might be, good follow study later on with a lot of billions of dollars in your
Yeah,
research.
that's a great suggestion.
Yeah.
I've never thought of to contact the pharmaceutical companies. Probably they have some data already. Yeah.
Yeah, they probably have, because as soon as you publish the paper, they probably start to do something, I guess. But, that might be good, next level of the study. And I believe that is kind of good way to go
Yeah.
further and then getting a lot of money, I guess. Uh, the let me check. And then the other one is, I mean, for second part, I fascinated by the the AlphaFold, and I start to use a lot of starting around 17 or 18, or even during the pandemic. I heavily used the AlphaFold and the Rosetta, because during that time we cannot do any experiments, so we shut down, and then we start to use those very extensively. So now you are combining AI, AlphaFold, all kind of fancy, prediction tools. I just wanted to ask, I mean, what is your view um, regarding those machine or AI in terms of the biotechnology in the future?
So for the AI machine learning, I think now we are having more and more data available, either through literature or from our own in-house data. So with more training data, I think machine learning and AI could be more and more precise in the future. But there is one challenge right now I have to mention is we have to have the consistent format for all the researchers if they want to deposit into database. That would ease our work a lot. Yeah. So yeah. And also there's a lot of models that's being developed, and I think they have a lot of progress from the CS part. Yeah.
Got it.
Just to add something quickly, I agree with what you just said. In general, in biology or bio-related fields like SynBio, bioengineering, there is a lack of standardization of many things, lack of standardization of the data, of the metadata, of the procedures. And we are all kind of, one way or another, limited by that.
Absolutely.
Yes, I strongly agree. That's why I said standardization. That's a better word. It's very important. Yeah. We deposit data onto database, but we need to make sure they are consistent.
Yeah, absolutely. So that remind me of my interesting discussion when I visited UC San Diego very recently to give a seminar. I met Terry Hua. He is the physics person originally, and then worked for the company for many years for the electronics I think company. And he said what we are doing right now with biology, we call it engineering biology. He doesn't believe we are doing engineering, because the one we are doing is, for example, in electrical system, if you can't connect, one device with the other device with a clear specification about how much resistance that have or how much capacity, capacitance they have, we exactly predict what will be final outcome, total, resistance or total whatever. We don't have that luxury yet. So that's
because we don't have any standards.
Yes, that's what I'm talking about.
And the point, we don't have standards. We have a hard time to define metrics.
Yeah.
And the big question, in fact, is it possible to define standards?
That is also a question
for biological system. I think it's possible at a certain level.
Yeah.
But I don't think right now we can push that idea that far, or it requires a lot of work, which is very interesting actually to think about.
Yeah, I completely agree with you, and that's my discussion with Terry Hua. He's the originally trained as the physics person, and he has very high standards in terms of the what is the engineering. And then the conclusion we made was we are still not doing engineering. We probably all doing art at this moment. That is the our conclusion. But I mean, we're getting there. We are one step by step. One day, I mean, who knows? I mean, AI potentially help us to make that happen. But we just hope, we just keep trying, and that's the one thing I want to tell. But one technical question to you. So you didn't show the data about the specificity of the engineered enzyme. You said this, change in this residue, have more, specificity toward the whatever compound, but those engineered enzyme also act on other substrate as well? Because that's also very
Yeah,
challenging.
so it does not completely switch the
Uh,
substrate specificity. It still has some effect towards the other substrate,
the
but
original
much,
substrate.
much worse. Yeah.
I see. That's my guess because I I done my protein engineering research when I was a graduate student. At the time, we didn't have the AI. I'm talking about 20 years ago. We didn't have the AI. We just, based on hardcore molecular simulation. And then I became a robot because at the time we don't have the liquid handler. So I was robot to picking 20,000 colonies and then pipetting triplicate, 20,000 colony, multiple time. And then we found some enzyme that's even better, but still original substrate, still kind of has some activity. So that's why I wonder, whether that completely changed, like, activity.
Yeah, that's true. But I think so. What I showed is just single mutations. So
Please.
what if we combine those single mutations together?
Yeah.
Maybe it could
Maybe.
switch
Yeah,
the
maybe.
specificity. Yeah.
Yeah. I mean, we recently show, I mean, that might be possible. We actually made a multiple very specific sensor, but this is sensor, not the enzyme. Enzyme is more trickier, because that one require docking and then conversion at the same time. Sensor is only docking simulation, probably solve the problem, but enzymatic reaction is docking first and then the conversion next. So that means even if you just focus on docking, I mean that doesn't mean
Yeah.
anything because that doesn't
Yeah.
tell you the conversion. That's KCAT. You need to talk about KCAT as well as
Yeah.
the other
There's
one.
two steps.
Thank you, Vincent.
Hi, everybody. I have to go. I have a workshop starting just in the 2 minutes. Yeah, thank you
Sure.
so much.
Thank you so much. I mean, thank you so much for your insight about the last discussion and also your early discussion.
Thank you.
All right. Thank you.
Thank you.
Have a good day. Bye.
Bye.
Okay. Let me close. Because we now see the 3 minutes before the 11th, the perfect timing. And then we chat afterward. So thank you all for joining and staying today. We'll meet again on February 6th, Thursday, the same time, the same Zoom link. We'll have Dr. Alex Munro Clark, an editor at Trends in Biotechnology, SynBYSS, and Professor Mariam Samadeh at University of Nevada, Reno. As usual, the following informal chat will occur without recording. Please stay here if you are interested with chatting with us. And thanks. I stop recording. Just give me 1 second.